Dear students, welcome to my channel. I hope you all are doing well. In my previous video, I discussed tissues, their types and functions. Today we will study anatomy of dicot and monocot stem, the difference between dicot and monocot stem, and different type of vascular bundles. Before we start anatomical study, I will tell you some morphological differences in dicot and monocot plants also. Monocots are differ from dicots in four distinct morphological features. हम मोनोकॉट को डाइकॉट से लीव्स के बेस पर, स्टेम के बेस पर, रूट्स के बेस पर और फ्लावर्स के बेस पर डिफरेंशिएट कर सकते हैं. बट द डिफरेंसेस स्टार्ट फ्रॉम द वेरी बिगिनिंग ऑफ द प्लांट लाइफ साइकिल इनसाइड द सीड at embryo stage. In the case of monocots, only one cotyledon is found, and in the case of dicot, two cotyledons. Cotyledons का मतलब होता है embryonal leaves. So, in the case of dicots, two embryonal leaves are found. और अगर रूट की बात करें तो in the case of monocots, fibrous root system is found and which is always found on the upper level of the soil. And in comparison to dicot root, in dicot plants only taproot system is found, which is deep seated root system. And third thing, on the basis of the leaves, both monocots and dicots form different type of leaves. Monocots leaves are characterized by their parallel veins called parallel venation, while dicots form branching veins which form network of the vein and veinlets called reticulate venation. आप इजीली डाइकॉट और मोनोकॉट प्लांट को देख करके डिफरेंशिएट कर सकते हो. मैंगो लीफ को आप देखो और वीट लीफ को आप देखो तो वीट लीफ के अंदर या मेज लीफ के अंदर या ग्रासेस के अंदर जो वेरिएशन वेन और वेनलेट्स होती है वो पैरेलल होती है, एक दूसरे को क्रॉस नहीं करती हैं. लेकिन मैंगो के अंदर आप देखो एक नेटवर्क फॉर्म होता है, वो डाइकॉट. जिनके अंदर नेटवर्क फॉर्म होता है वो डाइकॉट प्लांट्स होते हैं, और जिनमें पैरेलल होती है, क्रॉसिंग नहीं होती है वेन्स की, उनको कहते हैं पैरेलल वेरिएशन. तो वो हमेशा मोनोकॉट्स के बीच में होता है. एक इम्पॉर्टेंट बात, फ्लावर के बेस पर अगर हम डिफरेंशिएट करें, फोर्थ डिफरेंस मैंने बताया मॉर्फोलॉजिकल बेसिस पर, फ्लावर्स के बेस पर. अगर हम फ्लावर्स के बेस पर डिफरेंशिएट करते हैं तो फ्लावर्स जो होते हैं, मोनोकॉट्स फ्लोरल पार्ट्स आर यूजुअली 3 और मल्टीपल ऑफ 3, मतलब एंड्रोशियम, कैलिक्स करोला, एंड्रोशियम, गाइनोशियम, दे आर 3 और मल्टीपल ऑफ 3 मतलब 6, 9, और this is called trimerous condition, whereas in dicot floral parts consist of multiple of 4 or 5 or multiple of 4 or 5. This condition is called tetramerous or pentamerous condition. So, morphologically we can differentiate monocot from dicot by seeing their leaves, root system, floral parts, and number of cotyledons and anatomy of the stem. Now we will study the anatomy of stem. First, dicot stem. Now this is the picture of dicot stem. Dicot stem, the outer covering of the outline of the dicot stem is always circular, and outermost layer is single layer epidermis, which is found outermost. This is outermost covering which perform the function of protection, and it is made up of it is uniseriate. एक ही layer होती है, it is always uniseriate, consists of parenchyma cells, and it is cuticularized. So outermost covering is cuticularized, uniseriate epidermis, and few multicellular hairs are also found in the case of dicot stem. These multicellular hairs are called trichomes. डाइकॉट स्टेम का जो मैं एग्जाम्पल आज ले रही हूँ वो हेलियंथस प्लांट को लेकर के हम चल रहे हैं. तो नेक्स्ट इज below the epidermis, hypodermis is found. Hypodermis is cholenchymatous. आप ये कह सकते हो कि cortex is differentiated into three parts: outer hypodermis, middle cortex, and inner cortex. So hypodermis is always cholenchymatous in the case of Dicot stem. It is followed by middle cortex, and inner cortex is always parenchymatous. Next layer is endodermis. Endodermis is single layered, which is filled with lots of starch grains. These starch, just due to the presence of these starch grains, this is also called starch sheath. And below the endodermis, pericycle is found. Pericycle in the case of Helianthus is multi-layered and parenchymatous with alternating patches of sclerenchyma. आप देख सकते हो इस पिक्चर में vascular bundles जो हैं, vascular bundles at the top of each vascular bundle, this sclerenchymatous cap of pericycle is present. So it is dimorphic, consists of parenchymatous as well as sclerenchymatous cells also. Both alternate to each other. Now, vascular bundles. Vascular bundles are arranged in ring. In the case of Dicot stem, this is the characteristic feature of Dicot stem. Vascular bundles are arranged in a ring and they are always conjoint, collateral, and dark and open. Conjoint when xylem and phloem both are found in the same radius, jointly present हो और एक ही radius पर present हो, उसको कहते हैं conjoint. Next one is collateral, when phloem is found towards the outer side का मतलब towards the epidermis, and when xylem towards the pith or towards the center is called collateral type of vascular bundle. And dark when the inside the xylem, metaxylem are the larger xylem and the small xylems are called protoxylem. When the protoxylems are found towards the pith, this condition is called dark condition. And fourth condition is open. Open type of vascular bundle का मतलब होता है when the cambium is present between the xylem and phloem. Such condition is called open type. This is characteristic feature of Dicot stem. Cambium is not present in monocot stem. That condition when the cambium is not present, that is called closed condition. So हम monocot में कहेंगे कि conjoint, collateral, and dark and closed type of vascular bundles are found. Next is pith. In the case of Dicot stem, large central parenchymatous pith is present with intercellular spaces. Example हम Helianthus का लेके चल रहे हैं. Now we will study this diagram. This diagram, outermost layer, you can see some hairs are also present towards the outer side of the epidermis. These are multicellular hairs called trichomes. Below the epidermis, below the cuticle, layer of cuticle, outermost covering is epidermis, which is consists of parenchymatous cells, and epidermis is always uniciliate, single layer है और parenchymatous cells की बनी हुई है. Next, below the epidermis, cortex is present. This cortex is actually hypodermis. Outermost cortex is called hypodermis, which is consist of cholenchymatous cells. So it is hypodermis, which is consist of cholenchyma. Below the cholenchymatous hypodermis, middle cortex is present. That is consist of chlorenchyma. In the chlorenchyma, some resin ducts are also found in the case of Helianthus. And the inner cortex is consist of parenchymatous cells. Below the cortex, wavy ring of endodermis is present, which is filled with starch grains. So it is also called starch sheath. And then below the endodermis, pericycle is present. Pericycle is consist of sclerenchymatous cells. So patch of sclerenchyma is present in each vascular bundle in the form of cap. And phloem, these are phloem, which is found towards the outer side between the xylem and phloem. Cambium is present, and xylem towards the inner side, and metaxylem, large xylems are called metaxylem, और छोटे वाले xylem को protoxylem कहते हैं. And the center portion is parenchymatous pith. In the case of Dicot stem, large central pith is found, and between the adjacent each vascular bundles, between the two vascular bundles, the space is present. That space is called प्राइमरी मेडुलरी रेस. सो हम जो स्टडी कर रहे हैं वो प्राइमरी एनाटॉमिकल फीचर्स डाइकॉट स्टेम के स्टडी कर रहे हैं. नाउ नेक्स्ट इज मोनोकॉट स्टेम. तो डाइकॉट स्टेम मैंने अभी आपको बताया. डाइकॉट स्टेम जो आपने अभी पढ़ा उसके अंदर रिंग में वेस्कुलर बंडल अरेंज्ड है, और वेस्कुलर बंडल का साइज भी आपने देखा यूनिफॉर्म है, कोई छोटे बड़े वेस्कुलर बंडल्स प्रेजेंट नहीं है, और लार्ज पिथ प्रेजेंट है. नेक्स्ट इज monocot stem. This is the picture of monocot stem. Outermost layer is epidermis, which is consists of parenchymatous parenchyma. It is outermost layer, single layer, and uniseriate है, and covered with thick layer of cuticle. Multicellular hairs, बहुत important feature. Multicellular hairs, trichomes are absent over here, और अगर कुछ स्टोमेटा है तो वो बहुत कम है numbers में. Next is epidermis is followed by hypodermis. Hypodermis is made up of sclerenchymatous cells. Sclerenchyma is always they are dead cells. So in the case of hypodermis, sclerenchymatous cells are present, which provide mechanical support to the stem. Next is ground tissues. After hypodermis, the total portion in monocot stem is ground tissues, and vascular bundles are scattered in the ground tissues. There is no any type of endodermis, cortex, pericycle and pith. इस तरीके के tissues का differentiation नहीं होता है इसके अंदर. Next, vascular bundles. This is vascular bundle of monocot stem. In monocot stem, vascular bundles are skull shaped, and they are always scattered in the ground tissues. And each vascular bundle is conjoined, collateral, and dark and closed. अभी मैंने आपको बताया इसके अंदर conjoined, collateral एंड डार्क तो होते हैं, लेकिन क्योंकि जाइलम और फ्लोएम के बीच में कैम्बियम नहीं होता है, इसलिए इसको क्लोज्ड कहते हैं. And each vascular bundle is surrounded by few layers of sclerenchyma. This sclerenchymatous covering is called bundle sheath. Next is xylem. Xylem actually inside the vascular bundle, xylem vessels are arranged in V shape or Y shape. और जो bifurcate arm Y की होती है या V की होती है, उसके ऊपर हमेशा metaxylem होते हैं. And protoxylem dissolve. These are protoxylem. Few protoxylem dissolve and form a cavity which is filled with water. That is called protoxylem lacuna or lysogenous cavity, which is filled with water. In the case of monocot stem, pith is not differentiated. Pith is not found. So phloem, this is phloem, xylem, and few protoxylem dissociate and form lysogenous cavity, and the outermost covering of the vascular bundle is bundle sheath. So this is the structure of monocot stem, in which vascular bundles are scattered in the ground tissues, and there is no differentiation of endodermis, pericycle, pith and proper cortex. And in monocot stem, phloem is consists of seed tubes elements, and companion cells, phloem parenchyma and phloem fibers are absent. Next, difference between monocot stem and dicot stem. And on the basis of hypodermis, in the case of dicot stem, hypodermis is always consist of collenchymatous cells, which are living cells actually. But in monocot stem, hypodermis is sclerenchyma, which is consist of sclerenchyma cells, and sclerenchyma cells are dead cells actually. And when we talk about the vascular bundles in Dicotte stem, vascular bundles are arranged in a ring. और जो vascular bundles arrange होते हैं ring में वो हमेशा same shape and size के होते हैं, तो वो uniform in size, and they are open because between the xylem and phloem cambium is present, and bundle sheath absent, which surround the bundle sheath is the covering which surround the vascular bundle, which is absent in the case of Dicotte stem. But in the case of monocot stem, scattered vascular bundles are found which are scattered in ground tissues, and the size of the vascular tissues are not uniform towards the periphery. They are smaller in size. In the middle portion they are bigger, and the biggest type of vascular bundles are found in the center. तो जो size होता है वो vary करता है, और they are closed क्योंकि xylem और phloem के बीच में cambium नहीं present होता है, इसलिए इसको क्लोज्ड बोलते हैं. और वैस्कुलर जो बंडल शीथ प्रेजेंट होती इनके आउटर कवरिंग जो होती है, ईच वैस्कुलर बंडल इज सराउंडेड बाय sclerenchyma sheath, that is called bundle sheath, which is always found in monocot stem, not in Dicotte stem. And when we talk about the pith is present, large pith is present in the case of Dicotte stem, but it is absent in monocot stem. Medullary rays are present in primary medullary rays are present in the case of Dicotte stem, but it is absent in monocot stem. And secondary growth Dicotte stem में present होती है क्योंकि उसमें cambium present होता है, तो cambium become active in the case which is found between the xylem and phloem, become active and cut the cell towards both the sides xylem and phloem, and increase the girth of the stem. But it is this process is absent as are found in monocot stem. Next, concentric amphivessel type of vascular tissues. In such difference between dicot and monocot stem on the basis of the diagram. अब आपको इजीली अच्छे से समझ में आ जाएगा कि डाइकॉट स्टेम, ये डाइकॉट स्टेम है और ये मोनोकॉट स्टेम है. सबसे पहले हाइपोडर्मिस जो एपिडर्मिस के नीचे होती है वो कोलेनकाइमा की बनी हुई होती है डाइकॉट स्टेम में, और मोनोकॉट स्टेम में ये स्क्लेरेनकाइमा की बनी हुई होती है जो कि डेड सेल्स होते हैं. एंड अब वेस्कुलर टिश्यूज को आप देखो, वेस्कुलर टिश्यूज जो हैं वो रिंग में, वेस्कुलर बंडल्स रिंग में अरेंज हैं. इसके अंदर वेस्कुलर बंडल्स स्कैटर्ड हैं ग्राउंड टिश्यूज के अंदर. यहाँ पर हाइपोडर्मिस के बाद कॉर्टेक्स, एंडोडर्मिस, पेरिसाइकिल, पिथ प्रेजेंट है. बट मोनोकॉट स्टेम में आफ्टर हाइपोडर्मिस सिर्फ ग्राउंड टिश्यूज प्रेजेंट होते हैं, और ग्राउंड टिश्यूज के अंदर ग्राउंड टिश्यूज के अंदर वेस्कुलर टिश्यूज जो वेस्कुलर बंडल्स जो होते हैं वो स्कैटर्ड होते हैं. अब इसके अलावा जो है सेकेंडरी ग्रोथ प्रेजेंट होती है. वेस्कुलर टिश्यूज की अगर हम बात करें तो वी शेप इसके अंदर आप देखो, वी शेप जाइलम प्रेजेंट है, इसमें सिंपल जाइलम प्रेजेंट है. और ये जो है एक लाइसिजिनस वाटर कैविटी after dissociation of protoxylem it is produced. So water cavity is present in the case of monocot stem, but यहाँ पर कोई water cavity present नहीं है. और पेरिसाइकिल यहाँ पे स्क्लेरेनकाइमेटस कैप की फॉर्म में पेरिसाइकिल प्रेजेंट है, जो कि यहाँ पर एंडोडर्मिस भी प्रेजेंट नहीं है, यहाँ पर एंडोडर्मिस और स्टार्च शीथ इज प्रेजेंट. सो दीज आर द डिफरेंस बिटवीन द टू. पिथ यहाँ पर नहीं है, यहाँ पर लार्ज पिथ प्रेजेंट है. वेस्कुलर टिश्यूज का, वेस्कुलर बंडल्स का साइज भी आप देखो, यहाँ पर यूनिफॉर्म साइज के हैं, जबकि यहाँ पर बाहर की तरफ छोटे वेस्कुलर बंडल हैं, मिडिल में थोड़े बड़े, और सेंटर में सबसे बड़े साइज के वेस्कुलर बंडल प्रेजेंट है. नेक्स्ट, फ्यू टर्म्स जो कि आपने अभी यहाँ पढ़े वो मैं एक बार आपको और समझा देती हूँ. वेस्कुलर टाइप्स ऑफ वेस्कुलर बंडल्स. फर्स्ट जो है conjoined collateral and open type of vascular bundles. Conjoined collateral open type of vascular bundle. This type of vascular bundle, in this case, when the phloem and xylem both are found on the same radius, that is called conjoint. Collateral when phloem is found towards the outer side, means towards the epidermis, and xylem found towards the center or towards the pith. It is called collateral and open when between the xylem and phloem this cambium ring is present. Cambium is present between the xylem and phloem. This is called open type of vascular bundle. Conjoined collateral and open type of vascular bundles are found in the dicot and monocot stem. Next, conjoined and closed. Again conjoined, both are found xylem and phloem on the same radius. Closed when between the xylem and phloem cambium is not present. आप देखो यहाँ पर बीच में जो यहाँ पर येलो कैम्बियम बनाया हुआ है यहाँ पर एब्सेंट है, तो जब xylem और phloem के बीच में कैम्बियम एब्सेंट हो तो उसको कहेंगे हम क्लोज्ड. नंबर नेक्स्ट, conjoined and bicollateral. Conjoined and closed is found in the case of monocot stem. Open in dicot stem and closed in monocot stem. Conjoined, again xylem phloem on same radius. Bicollateral when xylem is surrounded by from both the side phloem, outer phloem and inner phloem. Such type of vascular bundles are called bicollateral. They are found in Cucurbitaceae, member of the Cucurbitaceae family. Type of vascular bundles. Phloem is surrounded by xylem, and example Yucca and Dracena, in which concentric amphi-vessel type of vascular bundles are found. Next is radial type of arrangement. Radial type of arrangement is found in dicot and monocot root. In such type of vascular bundles, xylem and phloem both are found on different radii and they alternate to each other. मतलब xylem alternate to phloem and phloem again alternate to xylem, और दोनों अलग अलग रेडियाई पर होते हैं. इसको कहते हैं रेडियल टाइप ऑफ वेस्कुलर बंडल. सच टाइप ऑफ वेस्कुलर बंडल्स आर फाउंड इन द केस ऑफ dicot and monopod roots. So this is the end of my lecture. Today you studied internal structure of helianthus and maize stem, their differences and different types of vascular bundles. So thanks for watching me. Stay tuned for my next lecture. Till then, goodbye.